Hello, good morning. Let's get started. Today we'll be doing lecture 8, digestive system study guide. That one should not take too long, probably like half an hour or so. And then we're gonna take up the lab and we'll do some review for test number 2 as well. So here we go. First question, list 4 accessory organs in the digestive system. So from the mouth to the anus, that's called the alimentary canal. And along the way, there are various accessory organs that will be adding secretions to the alimentary canal. And there are 4 accessory organs in total. Okay, so we have our salivary gland. So there are a total of 3 salivary glands. There is the parietal salivary gland, there is the submandibular salivary gland, and also there is the submaxilla salivary glands. We will label them later in the diagram. Another accessory organ, anybody can help me out here? What's another accessory organ?
Gallbladder.
Gallbladder is correct. Gallbladder.
Liver.
The liver is
liver and pancreas.
Yes, thank you all. Here are all the accessory organs. Okay, so liver produces bile, for example, and it's stored in the gallbladder, which would then get secreted into the small intestine, the duodenum. Okay, same thing for the pancreas. It makes the secretions into the duodenum through the pancreatic duct. Okay, so the food does not enter the digestive system, the food does not enter the accessory organs, but these accessory organs are important for the process of digestion. Okay, 4 layers of the alimentary canal. So we could think about the alimentary canal as a tube, basically, and then from the most superficial layer, to the deepest layer. Okay, so superficial is the outermost layer, and that is the serosa. Okay, serosa. I mean, serosa is just kind of holds everything in place. It has the serous fluid that lubricates the surface. So when your intestinal tract moves, for example, the fluid is going to reduce the friction, right? Because they will rub against each other, right? And by having a little bit of lubricant, it minimizes friction. Beneath the serosa is the muscularis. This is the layer that is going to have the smooth muscles. Okay, so smooth muscles are muscles that we do not have voluntary control over, and it's under control, the control of ANS, right? Your autonomic nervous system, and because this is related to digestive system, it is going to be controlled by the parasympathetic system. Okay, under the ANS. Right? Remember, the parasympathetic system is most active when you are relaxed, when you are in a low stress state, right? So under those circumstances, it's a good time to digest your food. So your muscularis layer is going to be most active. So typically there are 2 layers for the muscularis for a majority of the alimentary canal. There is the longitudinal layer, okay, which means the fiber runs parallel to the canal, and they're responsible for these kind of motion. And then there is the circular muscle layer. Okay, and the circular muscle, it's basically ring. Like if you imagine this is the alimentary canal, this is the lumen, that's where the food is. So the circular muscle layer is going to be going around the canal like this, okay? Whereas the longitudinal is going to be running in this particular direction, okay? So collectively, the two layers is going to provide kind of like squeezing and pushing. That propels the food forward in the alimentary canal. Okay. The stomach, though, the stomach has an additional layer, right, called the oblique layer. Oblique means diagonal, and that kind of allows the twisting motion, which is what we want, because the twisting motion can help mix with the food. Okay, what's underneath the muscularis? Anybody? Underneath the muscularis.
Submucosa.
That's right. Thank you. Submucosa. Submucosa is the layer where you will find the blood vessels. So they supply nutrients to the alimentary canal. They also is going to help with the absorption, particularly in the latter part of the small intestine, right? The jejunum and the ileum. This is also where you will find lymphatic vessels as well as nerve endings. Finally, the deepest layer, the layer that is going to be in direct contact, with the food, that's going to be the mucosa. Very good. So along the alimentary canal, we want to make sure that food is going to be going in a single direction. We don't want them to come back or move backwards. So much like our circulatory system, we have valves there to control the movement of blood. In the digestive tract, we have sphincters, which will control the movement of the food. Right. So in the order that we're going to pass through them, the first one is the sorry, go ahead.
Lower esophagus.
Yeah, the lower esophageal sphincter. That is correct. But that is also called the cardiac sphincter. Okay, it's the same thing, cardiac sphincter. And now I'll write here, or the lower esophageal. Okay, they're the same thing, and that kind of is found at the boundary between the esophagus and the stomach. Okay, so the cardiac sphincter will open to allow the food to go into the stomach, but it will prevent the content of the stomach from coming back out. Okay, if you have a leaky cardiac sphincter, if it doesn't close properly, then the acidic content of the stomach can come up, and it will perhaps give you a little bit of a heartburn. And then the lower part of the stomach, the next one, is the pyloric sphincter. That's correct, pyloric sphincter. Okay, and that regulates the amount of food that will go into the duodenum. It kind of squirt a little bit of the chyme forward a little bit at a time. We don't want to overwhelm the duodenum with all these chyme, because we need to take time to neutralize them. Okay, so the pyloric sphincter will help regulate that. And then we'll go through the small intestine. And before you go from the small intestine to the large intestine, there is another sphincter, the ileocecal sphincter. Okay, so the ileocecal sphincter is between the ileum and the cecum. Okay, ileum is the last part of the small intestine. In the small intestine, we have the duodenum, the jejunum, and the ileum. Right? So the ileum is the final section that will connect with the first part of the large intestine, right? The cecum. So that's why it's called the ileocecal sphincter. And last but not least, we have the internal and external anal sphincter. Okay, so that regulates, when you go to the bathroom, like it regulates when you do that. So those are the major sphincters. Any question? So let's do some polls here. Let's see if we, sorry, I forgot to erase all these things. Okay, it's pretty straightforward stuff here. Let's just do it. Go ahead. First one here. Yep, all of you got it right. Pancreas is not an accessory organ. Pancreas is kind of like an appendage that branches off from the cecum, right? And that is actually what we call a vestigial organ, right? Because like it's like no longer sorry, GLE, no longer needed. People can live without the pancreas, the appendix actually. But you know, it's believed to have a little bit of immune function. Okay, let's try the next one. Yep. The nerve endings in the lymphatic vessels and blood vessels, they are found in the submucosa. Okay. This one we haven't, I mean, we mentioned the salivary glands, but let's see if you are able to do this. Okay. All of you got it right. It is the parotid. Okay, so just to show you a picture, to remind you where they are. Okay, so here is the parotid gland right next to the ear. This big thing here, that's the tongue. So underneath the tongue is the sublingual gland. Lingual means gland, right? Like if you're bilingual, you speak two tongues, right? Two languages, right? So that's the sublingual gland. And then even lower, there is the submandibular gland. The lower jawbone, right? This bone here, that forms your lower jaw. That's the mandible. So the gland is kind of, you know, sort of like underneath that, I guess. So submandibular gland. Okay, very good. Moving on. The chemical digestion of protein first begins where? Anybody remember that?
Jejunum?
Sorry, go ahead.
Would it be in the jejunum?
Jejunum? No, good try though. Good try though. Anyone else? Jejunum, by the way, does not do digestion. Okay, since you mentioned it, the small intestine has three parts, right? Again, the duodenum, the jejunum, and the ileum. So the duodenum is where we do the chemical digestion, and then jejunum and ileum are for absorption.
Yes.
Okay. Stephanie says a mouth with saliva. Not quite, but again, good try. Anyone else? Protein. The
stomach.
The stomach is correct. The stomach is correct. Okay. So in the stomach, maybe we should probably talk about this. I know I mentioned this on in the video already, but since some of you it seems that not a lot of people know the answer for this one, so maybe I'll quickly talk about it. Okay. So in your stomach, right, you have these gastric pits. Okay, like this is the inside of the stomach. If I draw the stomach, and you don't really have to draw it, you can just watch if you want. Okay, so this is the stomach, and then you have your acid here. Now, if I zoom in on this surface, it's actually very uneven. Okay, there's a lot of grooves in it, and we call these the gastric pits, right? The gastric pits. Okay, and then at the bottom of it, that's where you will have the gastric gland. Does everybody understand the the orientation of the picture, like where we are and what I'm drawing? Any questions about that? Okay, so in the gastric glands and in the pit, there are different cell types. Okay, so there are these pink cells, for example. Okay, so these pink cells, we will call them the chief cells. Chief cells. Okay, so the chief cells, they are going to secrete something called pepsinogen. Okay, pepsinogen. So they will secrete pepsinogen. Pepsinogen. Okay. I don't know if you remember this, but this was like way back in lecture 2 or something, right? I told you ogen means inactive, right? So at the time we learned about like fibrinogen, right? So like ogen is inactive. So pepsinogen here is inactive, it's not going to be able to do anything yet. Okay, so to activate it, we are actually going to need something else. Okay, so there's going to be like these other cells, okay, in the gastric pit. These are parietal cells. Parietal cells. Okay, so the parietal cells will secrete hydrochloric acid. Hydrochloric acid. Hydrochloric acid, HCl. Okay, and what the HCl does is it will convert the pepsinogen into pepsin. Okay, it will convert pepsinogen into pepsin. So when you eat a protein, right? Protein, you probably should know by now, is going to be a bunch of amino acids, and then it's going to be all folded up like this, right? Okay, all folded up. So this is some kind of protein. And then what happens is when it gets to the stomach, okay, the hydrochloric acid from the parietal cell is going to denature the protein. It's going to unfold it. It's going to unravel it, okay, so that it becomes a long strand just with its primary structures. And then what's going to happen is the pepsin, which is an enzyme, is going to chop it up. Right? So it's going to chop it up into small strands of amino acids or individual amino acid. Okay, so this is the first part in the entire digestive system where your protein is going to be chemically digested. Okay, in the saliva there is no there's no acid in there, right? So it's not going to unfold the protein. There is no enzymes that would break down the protein. Okay, so it's the answer is the stomach. Is that okay?
I mean, I think the way you framed the question, because you asked where does digestion start, doesn't saliva break down starch?
Starch is not a protein though, right?
Yeah, no, but you, sorry, you didn't ask a protein. I thought you just said the question was, where does digestion begin? I didn't hear anything about a protein.
Oh, sorry. But the question here is maybe I read it wrong. The chemical digestions of protein, right? First begins.
Okay,
sorry about that. Okay, next one. Chemical digestion of carbohydrates. Where does that first begin? Anybody?
Mouth.
Now is the mouth. That's correct. This is for the mouth, right? Because there is an enzyme in the saliva that would break down carbohydrate. Anybody remember the name of that enzyme by chance?
Lipase. Amylase.
Amylase is correct. Okay, so protein first begin chemically digested in the stomach, for the carbohydrates in the mouth, and for the lipids, where would the lipids be first be broken down chemically in the digestive tract?
In the mouth.
We don't have lipase in the mouth, but good try.
And it ends in the small intestine.
That's right. So the duodenum. Okay, duodenum. Okay. So state a function of each of the following enzyme. So amylase is going to break down amylose. Amylose is found in starch, right? So amylose is going to be broken. The amylase is going to break down amylose into perhaps like some maltose, okay, or some glucose. It's hard to really write it in a chemical equation because amylose is basically many glucose together, right? So in the mouth, you're not gonna necessarily have enough time to chop up the amylose into individual glucose, right? You might not even be able to do it, to chop it into maltose, but you might be able to break it down into some we call like oligosaccharide, right? Like short chains of sugar. Okay, but the point is you're gonna start breaking down the amylose. Okay. Pepsin is gonna break down proteins into short strands of amino acids, right? Plus maybe some individual amino acid as well. Okay, like the picture I showed you, right? Think of the very long chain of circles, and then you just kind of randomly chop it in different places. Okay. Trypsin. Trypsin is the same thing. Okay. Trypsin is related to pepsin. They are cousins, chemically related. The kind of the difference is that pepsin you will find in the stomach and the pancreatic juice, but the trypsin is only found in the pancreatic juice. No trypsin in the stomach. Lipase is going to take lipids and you're going to break it down into glycerol plus fatty acids. Okay. So those are the enzymes. List 3 functions of hydrochloric acid found in the stomach. Right, so what are some functions? I already kind of told you. It will help you denature proteins. Okay, and what this does is it will make it easier, for pepsin to break them down. Okay, it's more accessible, right? You create more surface area for the enzyme to work on it. What's another function? Yep, Hafsa said eliminates bacteria. So good. It's like an immune defense, right? Immune defense. Okay, it'll break down bacteria, other pathogens. And one last one. Anybody remember?
It converts pepsinogen to pepsin.
That's right. Activate pepsinogen into pepsin. Okay. So the pH of HCl is about like 1.5 in the stomach, quite strong. And of course, the stomach is not bothered by it because it is protected by the mucus layer secreted by the mucus cells, right? Okay, list 3 features that will increase the surface area in the small intestine. Okay, so we have these circular folds.
Villi and microvilli.
Yeah, and there is the villi and the microvilli. That's right. So the inside of your intestine is not smooth. Okay, it's like kind of folded up like this. Okay, and then if you zoom in onto one of these areas, you will see that there are like villi coming out like this, which further increases the surface area. And then if you zoom in on one of the villi, you will see that they are actually made up of cells that has more little hair on them. So these little hair, they're microvilli. These pink ones, they are villi. And then these are circular folds. I'll show you the microscope slide in a bit, and then you can see what they actually look like. Okay, let's do a few more polls. I think we don't need to do this. Okay, like the function of amylase, I just told you to is break down carbohydrates. But maybe we can try this one. We can try this one. Go ahead. Okay, anyone else? Yep. Again, most of you got it right. The correct answer is the stratified squamous, because there's a lot of physical abrasion that the esophagus is going to experience. Like when you swallow food, there's a lot of rubbing against the interior. We swallow hot food, cold food, acidic food, and a lot of chemical abrasion as well. So to protect the esophagus, we need to have stratified, which means multiple layers of flat cells. This is the most ideal type of epithelial tissue that will protect against wear and tear. Okay. Next one, let's see if you just remember. This is a simple factual question. Okay. Anyone else? Only have 5 responses so far. Okay. The correct answer is the parietal cells. It's just like a factual question, right? Something that you just have to memorize. There's no explanation for it. Okay. True or false? Let's do this. First one, chemical digestion of food involves digestive enzymes that links monomer to form polymers using hydrolysis. This is not true. Okay. When we do chemical digestions, it's the other way around. Okay, we're gonna break down, right? Break down polymers into monomers. Okay, using hydrolysis, you probably remember learning this last semester. Right? Hydrolysis is the addition of water to break a polymer. Okay, all these macromolecules that we digest, they are all examples of polymers, right? Pepsin is an example of an enzyme that breaks down protein. We know that is true. Right? We will first get pepsin from the chief cells in the pepsinogen, right? From chief cells in the stomach, and then we'll get more from the pancreatic juices. Next, amylase is an example of an enzyme that breaks down carbohydrates in the mouth. We also mentioned that earlier. It is true. The liver is an accessory organ that secretes bile into the jejunum that helps in lipid digestions. What do you guys think? True or false for that one? True or false? You can just type the answer. And if it's false, what is the problem?
Secret into duodenum.
The duodenum, that's right. So actually the liver is an accessory organ that secretes bile into the gallbladder, right? Gallbladder, which would then secrete it into the duodenum, right? Okay, unless you don't have a gallbladder, then the liver will secrete it directly into the duodenum. You know, you will store it in the gallbladder first. The epiglottis prevents food from entering the nasal cavity during swallowing. That is not true. Okay? What should it be instead of epiglottis? I guess you can change it to the epiglottis prevent food from entering the trachea during swallowing. That would also work. But what structure prevents food from entering the nasal cavity during swallowing? The thing that dangles at the back of the throat of your mouth, the uvula. That's correct. Uvula. Okay, different parts of the tongue is responsible for detecting a specific taste. That is false. Okay, you know that diagram that you might have seen before, like the front is like sweet and then thigh is like for salty or whatever. Right, that's not true. The entire tongue is responsible for detecting, you know, all the tastes. Some areas are more sensitive than others, but for the most part, it's able to detect all the tastes in all over the surface of the tongue. The pyloric sphincter prevents acid reflux. That's not true. It is the cardiac sphincter. Food is pushed down the esophagus by wave-like contractions called peristalsis. This is an involuntary contraction of smooth muscle. That is true. Okay, peristalsis are created by the muscularis layer, and it continues to propel the food forward. Small intestine is separated into three regions: colon, rectum, and anus. That is very wrong. You can change it to large intestine. Chyme is neutralized by pancreatic juices in the stomach, not in the stomach, in the duodenum. Okay, we don't want to neutralize them in the stomach. It's still doing its digestion, right? Duodenum is mainly responsible for nutrient absorption, while jejunum and ileum is mainly responsible for nutrient digestion. It's false. It should be the other way around. The other way around. Okay. Bile works by emulsifying fat which aids in their digestion. That's true. Emulsifying just means like breaking them down into smaller pieces. It's a physical breakdown. You're not chemically changing the fat. You're just making it into smaller droplets. Villi are found in the small intestine to increase the surface area. That is true. Okay. And I think that is it. Okay, I think that is it. Any questions so far?
Can you please zoom in, Professor? I want to check up.
Sure. There you go. Okay.
Done.
Yeah. Done. Okay, great. Fill in the blanks. The tube that connects. Oh, sorry, this is grammatically wrong. The tube that connects the mouth. I should probably fix that. The mouth and the anus. I think I meant to say the tube that begins at the mouth. Yeah, begins at the mouth and ends at the anus. That's called the alimentary canal. Okay, but you know, people usually just call it GI tract, right? That also works. The gastrointestinal tract. The innermost layer of the alimentary canal that is in direct contact with the food is the mucosa. Mucosa. The layer of the alimentary canal that contains blood vessels, lymphatic vessels, and nerves is called the submucosa. The hard palate, which is the roof of your mouth, it's formed by the maxilla
and palatine.
and the palatine. That's right. So maxilla is the anterior 2/3, and palatine is the posterior 1/3. The uvula is an extension of the soft palate. It seals the nasopharynx during swallowing, so food doesn't shoot out from your nose. Mastication is the act of chewing. The parotid gland is located next to the ear. Amylase is an enzyme that is found, oh my goodness, so many typos here, that is found in saliva, which breaks down amylose. You will also find amylase in the pancreatic juice. A bolus is a ball of food. Peristalsis are wave-like contractions along the GI tract that propels food forward. The three layers of the muscularis in the stomach, they are the longitudinal, which is the outermost layer, circular, and then the oblique. Okay. Something or folds found in stomach lining when it's empty. Those are the rugae. Something is the hole in the diaphragm where the esophagus passes through. That's the esophageal hiatus. Next, something refers to thick, acidic, soapy liquid that leaves the stomach. That's the chyme. It's like a partially digested food, right? Mixed with the acid and all that. It's basically vomit, right? Pepsinogen is activated by HCl into pepsin. Intrinsic factor, which is produced by the parietal cells. So parietal cells makes the HCl as well as intrinsic factor. And it's a sugary protein that protects B12. Okay? If we don't have intrinsic factor, all the B12 is going to be broken down in the stomach. Then we're going to have B12 deficiency. Okay? And we need B12 to make healthy red cells with good hemoglobin. So people who don't have intrinsic factors because of genetic defects, they will get a form of anemia, okay, called pernicious anemia. 3 parts of small intestine: duodenum, jejunum, and ileum. Most of the macromolecules are broken down into their monomers, and the monomers are then absorbed directly into the bloodstream. But lipids, because they're not going to mix well with water, okay, because of their hydrophobic nature, so they're going to be absorbed through the lacteal, which takes them to the liver first, and then in the liver they will get combined with these lipoproteins. Right, so they will form like HDL and LDL, and then they will be able to go into your bloodstream. Bile causes lipid to break down into smaller droplets. We call this emulsification. Okay. The liver can convert glycerol into glucose in a process called gluconeogenesis. Gluconeogenesis. Okay, it's like creation of new glucose. And finally, we have the sodium bicarbonate, which is a buffering component found in pancreatic juice. So that sodium bicarbonate will go into the duodenum, and then it will neutralize the hydrochloric acid in the chyme. Okay, we need to do that because the duodenum does not have the mucus protection that the stomach does, and we need to protect the lining by neutralizing the acid. So that is that. Okay. These are pretty straightforward questions. Does anybody have anything you want me to go through? Okay. Application question. A patient has esophageal cancer and must have a feeding tube inserted. The nurse tells the patient that the tube will be inserted surgically into the duodenum. The patient's wife asks why the tube will not be inserted in the stomach. What should the nurse say? Anybody want to comment on that?
Is that because the digestion, that's where the digestion begins?
Yeah, that could be part of it, but why not put it in the stomach? I mean, we have all these acid in the stomach. Wouldn't it make more sense to put it in the stomach instead of the duodenum? I think you hit on one of the points, Colleen.
But the food would not actually break down that fast?
The food would, sorry, say that one more time.
The food will not break down as fast as it would in the stomach?
Okay, interesting. I'll definitely also mention another point that is very good. The acid would damage the tube. You know what? I actually never thought of that, but you are absolutely right. I think that that is another reason. Very good. But back to your point for a second, Colleen. On a normal person, do you think the food spend more time in the stomach or spend more time in the duodenum?
In the stomach?
Yeah, I would agree with you, right? Like you feel the food in your stomach sitting there for quite long, right? And the acid actually helps digest the food and break it down into smaller chunks, right? Like, when we swallow the food, it's like big chunks of, I don't know, meat, vegetable, whatever. And that acid really breaks it down into like a soapy substance, right? And then it will move to the duodenum where the enzymes can work on it. I mean, the enzymes cannot break down like a chunk of meat, right? If it's in the meat form, the acid really breaks it down into like a soapy form for the enzymes to work on it, right? And so if you think about someone who is receiving a feeding tube, what are we injecting into the person, right, through the feeding tube? Are we injecting solid or what do you think? Right? I think the answer is obvious, right? When someone's eating through a feeding tube, it's going to come in like a liquid pouch, right? The nutrient pouch, right? So because it's primarily liquid, there's no need to go into the stomach. Okay, the stomach's main job is to break down solid food into more like a liquid state. But if you're already injecting the liquid intake of the nutrient package, then it could go directly into the duodenum. Okay, so a couple of things, right? Number one, I like Stephanie's point of saying, the acid would damage your tube, okay, over time, perhaps, right? Increase wear and tear. Number two, we don't need to go through the stomach because we are intaking a liquid, right? And number three, the duodenum is where the chemical digestion is going to take place. Okay, it's also more accessible, right? Because it's like at the beginning of the small intestine, right? So it makes sense to insert it there. You just ate a large poutine for lunch. Use correct terminologies in anatomy. Describe how the poutine will be digested and absorbed in the digestive system. This is quite straightforward. I don't think we need to go through it. but the poutine is gonna have like what are French fries? What kind of nutrient is the wrong word, but like what kind of macromolecules is in the French fries? Anybody?
The carbohydrate and
Carbohydrates, exactly, right?
fat
So-
means lipid.
That's right. So carbohydrates and lipids, right? And if you put cheese on your poutine, which actually usually is the case, then you would have the proteins as well. Okay, so basically this question is asking you how we break down these things, right? So carbohydrates, we start breaking it down in the mouth, right, with the saliva, and so on and so forth. I think you will be able to describe this just by looking at the notes. Okay, next one. Chronic inflammation of the pancreas or pancreatitis impairs the digestive function of this organ, of the pancreas. Okay, so what would be the effects on digestion and absorption of carbohydrates, fat, and proteins? Well, the pancreas is going to secrete a mixture of digestive enzymes. So if you're not going to be able to do that, then the carbohydrates will not be fully broken down. Okay, the fat will be emulsified by the bile, but it won't be broken down. Same thing with the protein, right? But you know, some of the proteins might be broken down by the pepsin in the stomach, but you really have to rely on the pancreatic juice to do that. Okay, so if the pancreas is not secreting those enzymes, then you might not be able to absorb everything. Okay, much of these things, if it's not broken down into its monomer form, will just pass through your system, because in order to absorb them, you need to have the right receptor for it, right? So we're not going to have a receptor that's able to absorb you know, maybe 5 or 6 amino acid linked together. Okay, so that would just pass through your digestive system. Okay, and you might have a little bit of a malnutrition situation going on. Also, if you don't have the pancreatic juice, you might not be neutralizing the chyme properly. So you might have a little bit of a duodenal ulcer going on, right? You might irritate the lining of your duodenum, and that certainly could hurt. And that is it for the digestive system. It's really quite easy compared to the brain. If you survive the brain lecture, this is going to be you know, relatively easy. I think I have some harder poll questions. Let me see if we can do this. Okay, so there is a harder one that's probably worth your effort. So go ahead and try that. Okay, I only have 3 answers so far. Just gonna wait a little longer. Anyone else? Okay. We have 3 equally chosen answer, popular answers. So let's see what this means. Okay. If the baby constantly vomits, okay, so you're losing a lot of acid, right? Basically, you're losing HCl, okay, because you keep on vomiting it out, right? Okay. So in order to make more HCl, where do you think the H comes from? Okay, HCl, like if I want to make HCl, where does the H plus comes from? H plus comes from where? I know we said that the parietal cell is going to make the HCl, but where do you think the H plus come from in the body? Comes from the blood, right? Okay. So you're going to have to use a lot of the H plus from the blood to replenish the HCl. And if you lose H plus in the blood, then it will lead to alkalosis. Okay, alkalosis, which means there's going to be like a high pH. Okay. Oh, so I actually realized that this is a typo here. That should mean like high pH, low H plus. Maybe that's why it's confusing for some of you. Sorry. And acidosis means low pH and high H plus. Okay. So just to clarify again, in this situation, because the baby is losing a lot of acid and we need to replace the acid. Okay, so by making more acid, you are actually taking H plus from your blood, and that will cause your blood pH to rise, and you will have alkalosis. Okay, alkalosis. So that's it. That's it for the digestive system. Any questions before we move on to taking up the lab?
Yeah, I have a question. Just
Yes.
there is a question, but not related to the GI tract.
Okay.
Because actually I have to leave after some time.
Sure.
So the test will be, you wrote that 6, 7, 8 means special senses and the nervous system second part and the GI tract. But
That's
what
right.
about the lab? Lab will cover which area? Only
The
nervous
lab
too?
the lab is everything, right? The lab is 5, 6, 7, 8. Yeah.
Oh, this 5, 6, 7, 8. Okay.
That's
thank
right.
you.
Okay, thank you. Okay, let's do the lab then. Okay, here is the lab. There we go. Okay, so let me go full screen. Okay, first one. Identify the lobes A, B, C, and D. So A here. Oops. I'm just not sure why my pen is not showing up. Give me a second. Pen. Okay, so A.
Frontal.
Yeah, that is the frontal lobe. B, that's the parietal. Okay. C, occipital.
C, occipital.
Mhm.
D, temporal.
D is temporal. Okay. Lobe. What sensory information are processed by B, C, and D? So for B, the parietal lobe is processed somatosensory, bodily senses. Okay, so like things like pressure, temperature, pain, that's processed in the parietal lobe. What about C? What information gets processed in C, in the occipital lobe? Sorry?
Photorespiratory. Photo.
Yep. What kind of sensory information for the occipital lobe?
Visual.
Visual, that's right. Visual. Right. And for D, for temporal lobe, we have auditory, which is sound, right? And olfactory, which is smell. Right. Just let me know if I'm going too fast. Again, I'm assuming that you have done this already. Next one, label E, F, G. So E is cerebellum. Okay, what kind of memories are stored in E? It is the skill memories. Skill memories. You know, motor skills, right? Playing the piano, riding the bike, typing on the keyboard. F is the pons, and G is the medulla oblongata. Okay. Label the functional cortex H and I. So this is the front of the brain, and this is the back. So in the frontal lobe, oh, please go back, Hafsa said. There you go. Okay. So here at the back of your frontal lobe, right, this area, this is the primary motor cortex. Okay, so it allows voluntary movements, right? Anytime you move any parts of the body, you will need to originate that command from the primary motor cortex. Okay? And of course, it controls the opposite side of the body. Okay? So this here, we're looking at the right half of the brain. This will control movements of left half of the body. This is the primary somatosensory cortex. Okay. I should maybe mention this, that the this area here, okay, so this groove, right? This groove here, that groove is called the central sulcus. Okay, like this groove right here. So this is the pre-central gyrus, and this is the post-central gyrus. On the pre-central gyrus, that's where you will find the primary motor cortex. Okay, so it seems like it's pointing at the same thing, but then the gyrus is just kind of referring to the physical location of the brain, and then this is referring to the functional area that is found in that region of the brain. Okay. So on the test, if I ask for the functional cortex, then these would be the answer. If I ask for the name of that region, I don't think I do ask you that, but if I do have a question that asks you what is the region, this region of the brain, then that's the pre-central gyrus, right? Okay, so the pre-central gyrus contains the primary motor cortex. Okay, and then the post-central gyrus contains the primary somatosensory cortex. Okay, so again, the function of the somatosensory cortex is to process somatosensory inputs, right, from your skin, touch, temperature, texture, all the things we talked about. Is this okay? Any question? A is the corpus callosum. It's white matter and it enables communication between the hemispheres, the two halves of the brain. What is B here? Anybody got that one?
Thalamus.
That is a thalamus. Very good. Which filters sensory information. Except for smell. Before projecting them to the appropriate lobe. Okay, so if it's visual information, it's going to go to the occipital lobe. If it's somatosensory information, it's going to go to the parietal lobe. Okay.
Is B pointing to the little purple circle?
No, B is pointing at this whole thing. So what is that little purple circle? We didn't talk about it in class, but this purple circle basically allowed the right thalamus to communicate with the left thalamus.
Okay.
Yeah. I have another question. What is in front of the third? Sorry, I almost gave you the answer. What is in front of the thalamus right here? Okay, so this space in front of the thalamus, what is that part of?
Third ventricle.
That's correct, Mohammed. The third ventricle. Okay, remember the third ventricle is like this, and then the walls of the third ventricle is formed by the right and the left thalamus. Okay, if you picture, like if you're in a room right now, okay, if you picture you sitting in the thalamus in the third ventricle, right? If you picture you sitting in the third ventricle right now in your room, the right wall is the right thalamus and the left wall is the left thalamus. Okay, what about the floor? What would the floor be made up of? The hypothalamus. Okay, so if you picture the third ventricle as your room, you would have the thalami on each side and the hypothalamus at the bottom. Next one, C and D. Okay, so C is the, as I just mentioned, where is my pen? C is hypothalamus. Okay, so what it does is it maintains homeostasis, right, through hormones. Okay, and we will learn about all the hormones later on. How about D? What is D? This little thing that's dangling at the front.
Pituitary gland.
That is the pituitary gland. Very good. Pituitary gland. Okay, so also uses hormone. Uses hormone to regulate various process in the body. Okay, I'm gonna ask another question. What about this? What is this thing right here? Okay, there's a little white thing right here. Anybody know what that is? Oh, thank you, Joan. Very good. It is the optic chiasma. Okay, so that's where the optic nerve crosses the middle, right? On the way to the thalamus. Okay, so it's in front of the area that's between the hypothalamus and the pituitary gland. Very good. Okay, next one. Label E and G. So E here, this little triangle area. E is the fourth ventricle. And G is the central canal. Okay, which is in the middle of your spinal cord, right? Okay, this is a CT scan. Let's find everything we can. So corpus callosum is probably the easiest to identify. There we are. That is the corpus callosum. Cerebellum also quite easy to find. It's this thing right here. Cerebellum. Pons and medulla oblongata. Okay, right here. Okay. There's a request to go back to this one. Okay. Any question about this CT scan here? No? Okay. More. Okay. Lateral ventricle. Well, you cannot see the entire lateral ventricle, but here you can see a bit of it, right? That's the LV here, lateral ventricle. Right, just underneath the corpus callosum, but it's much bigger on the inside, right? Much of the lateral ventricle resides within the hemisphere, right? The third ventricle, it's in front of the thalamus right here. So that's the third ventricle. Third. Cerebral aqueduct, that's this duct right here. You see this line? Right? That's the cerebral aqueduct. And then this triangle, it's very hard to miss. That's the fourth ventricle. Okay, so it's located behind the pons and in front of the cerebellum. Okay. Anything else we can identify from this diagram? Anybody? Like for example, right? What is this here? What is that? What if I point to that on a test and ask you what it is? What are you going to say?
Pituitary gland.
Yep, it's the pituitary gland again. Okay, and again, this here, this area, that would be
Hypothalamus.
hypothalamus. Thank you. Okay. So as I mentioned before, it's a good idea to just look these things up on the Google image, then just try to label as many things as you can. It will definitely help you, remember things a little bit better. Okay, next one. List 3 functions of A. So A is this
Cerebrum.
So it's the cerebellum. Yep. So the function, it's like maintain posture. Right, maintain posture, store memory, skilled memories.
Coordinate movements.
Sorry?
Coordinate movements.
Yeah, coordinate movements. Right, ensure you have smooth, seamless, voluntary movement. Okay. Very good. Label B, C, D, and E. Okay, so B here is again the pituitary gland. Okay, there is the anterior and posterior pituitary gland. C is the frontal lobe. We're just looking at it from the bottom. These white things, right? These are actually the, these white things here. Those are your cranial nerves, which we didn't learn, but we know that there are 12 pairs, right? Okay, so those are the cranial nerves. D, which lobe is D from this perspective?
Temporal.
Temporal.
Temporal, yeah, very good. And E is again the medulla oblongata. Oblongata. Okay. So we're gonna take away all these things, this model, and we're gonna look at it without everything else. So what are we looking at for A? These spaces. What did you guys put for your lab?
Lateral ventricle.
Those are the lateral ventricle, yes. Lateral ventricle. Okay. Okay, we're looking at the left brain here, left hemisphere. Okay. Label the functional area A. Okay, so this, if you remember from class, it's the Broca's area. B is the Wernicke area. Wernicke area. So Broca's area allow you to have fluent articulate speech using the right words in the right sentence and the right context and with the right syntax, basically to speak properly. Wernicke's area allows you to understand, to interpret speech as well as written communication. What is missing at the question mark? What? Yeah, so usually this here, this is where the cerebellum goes. This here, that's the lateral sulcus or lateral fissure, depending which book you read. All right. So it separates the frontal lobe from the temporal lobe, basically.
So the central sulcus would be right at the top.
Yeah, this
ear A?
model does not have a central sulcus, but if I were to, visualize where it is, probably like somewhere like here.
Okay.
Yeah. The other one has a nice central sulcus like this one, right? This one you can see clearly separates the lobe, right? Like on the actual brain, you would actually see a groove. Okay. So to visualize where the central sulcus is, right? It's like, if you picture like where, like I'm wearing my headphone right now, but if you think about wearing like a hairband, this actually is where the central sulcus will be on the side. Okay. Okay, so it's the same brain, and then we took it. This brain costs like $400 to get at the school, but it has a lot of parts in it. So here, A again, that is the frontal lobe. Okay, I don't think we have to write that again. C is the temporal lobe. Okay, frontal, temporal. But what is B? This is the one a lot of students ask me on the in the lab. What is B right here? Jung said the thalamus. Good try. The thalamus would be on the more of the on the interior, on the medial side, right? On the medial side, if you look at it from the middle, right? Do I have a medial view? Yeah, like so the thalamus is more like here, right? And when you look at it from the middle. So anyone else want to try for B?
Would it be the cerebellum?
Cerebellum. Cerebellum is like here at the back, right?
Yeah, okay.
Yeah, like the big brown thing, right? Okay. Anyone else?
Thalamus.
Sorry? I just didn't hear, sorry. The insula. Remember in the, what is it? The sensory organs unit, right? I said if you actually, you know what? Let me see if I can show you. So if you stick your hand into the frontal, into the lateral sulcus like this, and then you pull it apart. You actually could do that, like with a real brain. You kind of grab it open, then you will be staring at the insula. Okay, that is the lobes for, amongst many things, it's responsible for processing taste, right? Gustatory information. Okay, once again, D is the lateral ventricle. Okay. Can you visualize what's going on here? Like this thing fits into here, right? And then this part would go to here, and then when you put it all together, it forms this brain, right? It's hard to see, I suppose, but that's what it is. Okay. Medial view, identify the following structures. Corpus callosum, you should have no problem identifying that by now. Okay, pineal gland. Pineal gland is right here, this little tiny gland. Pineal gland secretes melatonin. What do we need melatonin for?
Sleep.
That's right, to induce sleep. Okay. You're gonna feel tired if you have a lot of melatonin. Okay, 21, 22, and 23. 21 is just the thalamus. In front of the thalamus, again, is the third ventricle. We talked about that many times already. If you can find the thalamus, then below it is the hypothalamus, homeostasis, and then 23, pituitary gland. And again, 25 here, that's the pineal gland. Melatonin helps you sleep. Okay, label as many parts as you can and sketch the sensory neurons. I mean, we've done this several times already. So, I mean, we can try to sketch it again. We have the sensory neuron coming in, right, like this. So this is the sensory neuron, okay, which is the afferent pathway, right? Afferent pathway. Right. And then what's going to be in here?
Gray matter.
That is the interneuron. That's right. Interneuron. The gray matter. Right? So that's the interneuron. And then finally, that's going to make a connection with the motor neuron that will come out at the front. Okay, so that's the motor neuron, the efferent pathway. Okay. So what else do we know from here? What else can we label? A couple of things. This whole thing is like your spinal nerve, right? Spinal nerve here. This is the back, so that's the dorsal side, and this is the front, the ventral side. This is a particularly nice cut. You can see the meninges. Part of it anyways. I imagine that is the dura mater. This hole in the center, that is the central canal where the CSF is going to go through. What are we going to find at the back here? You're going to have
Ascending and descending tracts.
Yep, at the back is going to be the ascending tracts, right? So all these things going up to the brain, ascending tract. Okay, they are going to be taking sensory information to brain. Okay, and then the front, you would have all the descending tracts. Descending tract, which will be taking motor information. Motor information means like moving body parts, right? From brain. And like I said, the size is going to be like mixed. Okay. White matter on the outside, gray matter on the inside. Any question? Okay. Identify two types of cells. This was hard to tell, I guess, but this big thing here, that is your neuron. Okay. So you have like your dendrites kind of coming out. Okay, and then it's hard to tell which one is the axon. If I have to guess, maybe this one. It's going to continue. Okay. And all these here, these are the nuclei of glial cells. We cannot really make out the actual cell. It's not focused properly, but we can see all these nuclei, nuclei of glial cells. Okay, the supporting cells, right? Okay, well that took a long time, but that's all the brain stuff. Any question about the brains before we move on? I would think the brain is the hardest, right? Of the 3 units, probably requires the most memorization. Okay, we're just gonna skip the break, okay, and finish the review. Ear overview. A. What is this? This would be the auditory canal, the ear canal. Okay. It funnels sound towards the eardrum. B, these little tubes. Anybody can tell me what they are?
Semicircular canal.
That's right, semicircular canal. For detecting rotational motion of the body, right? Yeah. And C is the Eustachian tube. And the function is to equalize pressure within the head with the environment. Okay, so I'm just going to put equalize pressure with external environment. Okay. This is the inner ear, and you see a couple of things here. E is the cochlea. Cochlea for hearing, right? Inside is full of fluid. Identify the semicircular canal. Okay, so this one is the anterior. Okay, again, assuming you are in a room right now, okay? So your left wall and your right wall would be parallel to the anterior semicircular canal, okay? The roof, the ceiling rather, the ceiling and the ground would be parallel to the lateral semicircular canal. And then the wall in front of you, as well as behind you, that would be parallel to the posterior. Okay, so anterior will be stimulated if you nod your head. Lateral will be stimulated when you shake your head. And posterior will be stimulated when you tilt your head. Okay. Same thing for the entire body, right? Okay, if you rotate like this, that would be lateral stimulation. So there's fluid inside that will basically spin with your motion. D, that's another one that a lot of people don't usually get. What is D? Anybody? That is the vestibulocochlear nerve. Okay, so that takes signals from the vestibule, which is the part that connects the semicircular canal with the cochlea, as well as going to take information from the cochlea and then send it to the brain. F, down here, almost missed it. F, that's the tympanic membrane or eardrum. Tympanic membrane. Next one, middle ear, label XYZ. Z is again the tympanic membrane. The function of tympanic membrane is to convert sound energy into movements. X, which looks like a hammer, is the hammer or the malleus. Y, the one with the pointy thing, right? This is the incus or the anvil. And then there is also the stapes, but I think it broke off at the model at school, so I wasn't able to take a picture of it. But collectively, the malleus, the incus, and the stapes, they are gonna, so plus stapes, that's gonna equal to the ossicle. Okay, ossicles. I know it looks like it's pronounced stape, but it's actually stapes, for those of you who are wondering. We good? Any questions? Okay, so this is another model. Right? This one you can see clearly. This is the stapes, right? Okay, or stirrup. What does it connect to? Anybody? What does the stape press on?
Oval window.
Very good. Oval window, which would then transfer the movement into the fluid within the cochlea. But of course, every time the oval window gets pushed in, the round window, which is further away, will get pushed out so that you don't build up too much pressure in the ear. Okay, label as many parts as you can. Okay, so here we go. So that's the incus again, incus. That's the hammer. Sorry, I should stick with the proper term, the malleus. Okay, you can see a bit of the stapes here. Semicircular canal. The nerve that we talk about, right? The vestibulocochlear nerve. Let's just call it VC nerve for short. This shell here, that is the cochlea. Connecting the cochlea to these things. Vestibule, which is used for detecting acceleration, right? So you're speeding up, slowing down, falling. That's the vestibule. Yeah, so if you imagine going on a roller coaster, all that sensation that you feel, but going down, falling, or turning, loop the loop, all that sensation is picked up by vestibule as well as semicircular canal. Okay, so if you do, when you're doing like a loop the loop, right, like going up like this, right? It's the anterior semicircular canal that gets activated the most. All right. Anybody had any extra label that I missed? Yeah, I think that pretty much covers it. I mean, you can kind of see a little bit of the eardrum here, tympanic membrane, and then the auditory canal. Okay, we still have like a lot of slides. We're only on like 23 out of 39. So maybe we will take a 5-minute break just for you to freshen up a little bit. And then we will continue, okay? 13 is not something that we learn in class, so don't worry about it. It's just part of the bone structure. It's part of your temporal bone, actually, that helps form the ear canal. Okay, but let's take a break. Okay, we'll return in 5 minutes. Okay, let's keep going. Next one. Now we are on to the eyeballs. So A, that is sclera, which is the white part of the eye. It's a fibrous type of tissue, and it allows muscle to attach to it. So here you can see like an eye muscle right here, right? Eye muscle. Okay. There is also one at the bottom, on the other side, as well as on top. B is the choroid. It's the vascular layer of the eye. You can see all these blood vessels and you know, nerve endings. They will nourish the eyeball. C, that's the retina, where you will find the photoreceptors. Okay. If you open up the eyeball, you will see this big gel thing. What is that? Anybody can help me out here? What is that?
Is that the vicarious humor?
That's right, the vitreous humor.
Vitreous.
Vitreous humor, which is a gel-like substance that helps reinforce the shape of the eyeball. D. D is the cornea. State a function of E, which is that reinforce the shape of the eyeball. F, that is the optic nerve. Okay. What about G? Anybody know what G is pointing at? Not the blood vessel, but like this, you know, this overall area. Oops. What is that? Anybody? Okay. G is the blind spot. Blind spot, also known as the optic disc. Okay, so where the optic nerve exits, you cannot have photoreceptors on it. That's why it's called a blind spot. State the pathway. So we start with the optic nerve, which will go to the optic chiasma. And then it will become the optic tract once we are inside the central nervous system. We'll go to the thalamus. And then it will go to the occipital lobe. Remember, right field of view ends up in the left occipital lobe, and then the left FOV ends up in the right occipital lobe. All right? What about this tiny spot here? Please tell me what that is.
Is it fovea centralis?
Fovea?
Yeah, the fovea or fovea centralis, that's right. It has the highest density of cone cells in the retina, and it produces sharp image. Eye, this is the ciliary muscle, right? Which controls the tightness. We say tension, okay? The tension of suspensory ligament. Which in turn, of course, affects the thickness of the lens. To look far away, we want it to be a thin lens. To look close by reading a book, we want it to be a thicker lens. I know, it's tiring. Okay, next one.
Sorry.
What is, that's okay. What is found in number 2 right here? What is in that space? That is the aqueous humor.
Aqueous humor.
That's right, the funny water, the aqueous humor. J is the lens, and the clouding of J is it's called cataracts. Right, it's when the protein starts to crystallize in the lens, and then you will slowly lose peripheral vision. Okay, you can do surgery to fix that and get an artificial lens. Okay, that's it. That's it for the sensory organs. Okay, and in the lab we actually have these. It's pretty big. It's almost like this big, like of a human model, and that's what we're looking at right now. And we're going to try to label as many things as we can. Okay, so number 4, right? Number 4 right here. That is just the cerebrum. Please do not confuse cerebrum with cerebellum. Okay, number 5. Everybody should know number 5 now. We must have talked about this at least 4 times today. That is the corpus callosum. Seven is the pons. 8 is the spinal cord. It's not the medulla oblongata because we already like out of the head, right? Out of the brain, right? This is the This is the hole at the bottom of your skull that lets it come out. So anything that's out of that, right? This is actually the bones of the of your backbone of your vertebrae, and this is the spinal cord. Okay. 9 here is the nasal cavity. 10 is the tongue. Okay. And J shouldn't be here. J is like from here from the last slide, and then I forgot to erase it. So don't worry about J. 11. What's 11? Anybody know what 11 is? That's the maxilla. It forms your hard palate. Okay. Number 12. Just gonna put it here. It's getting too crowded. 12 is the soft palate. And number 13 is the extension of the soft palate, which is the uvula, which will go back and seal the nasopharynx when you swallow. 14 is another hard one that usually people don't get. Anybody have an answer for 14 before I tell you? Just wondering. We learned a long time ago. That's the adenoid tonsil. Okay, we learned this in lecture 4. Is it? No, lecture 2, when we do the immune system. Okay. That is that. Next, the thorax. Okay, so number 16 here, we have the thyroid cartilage. Number 19 is the superior lobe. I know this is like, last unit, but because we're doing the full body, that's why we have some extra labels. Okay, middle lobe. And then we have the inferior lobe. Okay. This here, that's the aorta. And that here is the pulmonary artery. Okay. Oh, and there is another one here. That is the thyroid gland. Moving down. This is the abdomen. What did I take out in this model? What's missing here in this big space? Anybody can tell me? What's missing there in that big gap? Okay, I took out the stomach here, okay? So normally this is where the stomach is. This is where the esophagus is. And then this is gonna go to the duodenum. And then you would have the stomach like sitting right here. Okay. So behind the stomach, we have number 35, which is pancreas. 41 is the transverse colon. 34, that's the jejunum. Okay, 42, it's in the picture, but it's hard to see, I guess. That is the descending colon. Okay. Couple other things that you might recognize. This thing right here, this structure. This right here. Anybody recognize what that is before I tell you? Again, just checking to see if you're with me. That is something we learned before. Oh, okay. Spleen. That's what it is. Okay. Stores blood, right? It's of course used to have a cumulative final exam. I'm sure you're glad that is not the case anymore. Okay. Familiarize yourself with different parts of the large intestine. This is all labeled. You just have to go through it yourself, right? Ascending, transverse, descending, sigmoid, so on and so forth. So I'm not going to talk about that. Stomach. Okay. A. Oh my goodness. A is the esophagus. The lower part anyways, right? Lower esophagus. B, that's where you will find the cardiac sphincter. Okay. C is the, main part of the stomach. And you can see all these foldings. Those are the rugae. Rugae shown. That's the pyloric sphincter. And E, duodenum. Okay. And when you're studying, you should write down the function of each of these things, right? Okay, accessory organs. A is the liver. B is the gallbladder. Pancreas. Duodenum. Okay. So secrete sodium bicarbonate and digestive enzymes. Into the duodenum. Right? Digestive enzymes will break down the macromolecules, and the sodium bicarbonate, it's going to neutralize the chyme. Okay, this is a esophagus, transverse section of the esophagus. So what you can see is.
Can you go back to that quick slide?
Yeah, sure.
Okay, thank you.
No problem. So here, this part here, this thin layer, that is the serosa. Okay, very thin. Okay, so like here, this one that goes all the way around. I can't really trace it, but you can see the fang halo, right? That's a serosa. And let me see if I can change the color. This here doesn't really show up too well. That's the muscularis. Okay, muscularis, right here. Okay, this part here. And you can see there are two layers, right? You have the longitudinal on the outside, and then you have the circular on the inside. Okay, so, this whole thing is the muscularis. And then this part, let me switch color again. This part here, this kind of like a purplish blue, I don't know what color to call that. That is the submucosa. That's where you'll find, again, the blood vessels, nerve endings. And then finally, the mucosa is the innermost layer. Okay, so let me switch back to red right here. This deep purple color layer. Okay, so on and so forth. That is the mucosa. Okay, and the inside here, that's the lumen, right? The inside lumen. That's where the food's gonna go. Any question about the 4 layers? Okay. Small intestine. Identify the 4 layers again. Okay, so again, it's easiest to go from the outermost layer. This is the serosa. Okay. You can see the muscularis here. Right, you have the outer longitudinal right here. That's a longitudinal. And then this is the circular. You can see that that's where the boundary is, right? You see these are blood vessels, okay, and veins. Okay? So that means this layer is the submucosa. And then finally, this is the mucosa. It's kind of cool in class if you are able to see this yourself. But now we can only see the pictures. Okay, this is another small intestine. So this whole thing, right? These things, that's the PC or circular folds. Circular folds. Okay, that's one of the structures that would increase the surface area. The ME here, it's the muscularis. L is a little bit tricky, but it's the thing that is in the middle of a villi and it helps you absorb fat. So that's a lacteal. V is pointing at these structures. Okay, that's V. That's the villus. Villus for singular, villi for plural. Sub-M, you probably guessed that one, is the submucosa. And this is the mucosa. Okay, you cannot really see the microvilli here. You would need a more powerful microscope to zoom in to see it. And finally, just one more. Which side is the surface of the tongue? Okay, so this here, that is a papillae. Papilla, and then plural is papillae, which means this would be the surface. Okay, that's where the food's gonna go. And the taste buds are these little things that are on the side. Okay, so if you look at the papilla on the side, that's where you will have the taste buds. Okay, taste buds. So these, lighter shades, those are the taste buds. Okay, that's it for the lab. So you will have 2 days to complete the lab. It's going to be available starting tomorrow at 6 a.m. At 10 a.m. rather, 10 a.m. And then you're going to have until Friday, 10 p.m. to complete it. 2 attempts. Highest score will be recorded. Okay. I already posted the information on eCentennial, but let's quickly go through it in case you need to see it. So obviously do the lab quiz before Friday. And the test review that I posted, you can work on that again. And then this is test 2 information, 75 minutes. It's going to be on Monday from 6 a.m. to 11:59 p.m. And here are the breakdown, and it's going to be another 20% of your final mark. Okay. Now, I don't think I will have time to go through the entire test review. So let's just go through what we can. All right. The test review, again, you can just click on this link and that's where you can download a copy. I mean, the first one is pretty self-explanatory. We already kind of did some of these things when we go through the lab, right? Just to label the brain and then states its functions. Pretty self-explanatory. And you know, whatever we did not cover, you can go through the lecture notes to find it. But it's very important that you fill this in. Okay, it's a good idea to even create this chart in your own way to summarize it. And maybe include a picture of where it is. That would be a very, very useful studying tool. Okay, so here, let's do this one. Complete the following chart for key functional areas of the brain. Okay, so the primary motor cortex. We talked about this multiple times already. And you know, you should be able to identify it. Okay, so here is the primary motor cortex, right? Okay. Will we have class on Monday for the test, or will this be done? This is the last class before the test, okay? So Monday you will have the test, and then Wednesday we will do the kidneys, okay? So it's a lot of stuff going on, right? After the test, make sure you also watch the kidney recording, because kidney is it's also very complicated. All right. So there it is. That's the primary motor cortex. And the function. What's the function of that? The function is, of course, to. I'm not going to write it again. You can write it when you study. We did it in the lab, right? The function is to enable voluntary control, a voluntary control of the other half of the body. Another key thing to remember for the primary motor cortex is that it is separated into different zones, right? Okay, so this part is going to control your face. This part's going to control your hand. And each section is responsible for a particular region in the body. Primary somatosensory cortex. Again, you should know by now. It's going to be here behind the central sulcus. And that's for somatosensory input. Okay, we also wrote it down during the lab. Broca's area, that's for, it's your motor speech area so that you can talk properly, right? With the articulate syntax and everything. And the Wernicke's area is here in the temporal lobe. Okay, so make sure you know where these areas are. It's not uncommon to, like on the test, I might point to a particular region. I can ask you straight up what it is. So you have to know the name of the region. I can ask you what happens if that area is damaged. And so make sure you know where they are, what they are called, and what happens if it's you know, damaged. Describe the anatomy of the spinal cord. We did that as well during the lab review. You touch a hard surface, describe the neuronal pathway involved in processing this information. Okay. So this has to do with drawing the spinal cord, you know, with the pathway coming in and going back out. We did that just now with the lab, but also going up through the ascending track, right? Okay, so sensory information is going to go up the ascending track. It's going to cross the midline at the medulla oblongata, right? It's going to go through the pons, the thalamus rather, and then eventually it's going to end up at your primary somatosensory cortex. So you want to be able to trace out that pathway and know exactly what each part does. Okay, so here are some examples. Okay, identify the indicator structures. Okay, I'm just going to tell you the answer. We don't have time to do the polls. So this is the corpus callosum. And next one, this here, the bulging part, that is the pons. Which of the following that is not a function of the indicator part, right? So something like this, not only do you have to know what this is, you also have to know what it does, right? So of course, by now you should know that this is the cerebellum, and cerebellum is going to store motor skill, is going to allow smooth execution of motion, maintain balance and posture. What it does not do is it does not maintain alertness and consciousness. What's the structure that does that? Maintain alertness and consciousness? Can someone tell me, please? No? What maintains alertness? Are you alert now? Consciousness? What part of the brain does that? That is the reticular formation. Okay, reticular formation. It's in the notes. Okay, take a look. This one, we just did it above. That's the primary motor cortex for voluntary movement. This one, what is it pointing at? It's pointing at these structures. This is a picture directly from your notes. It's the thalamus. Identify the indicator parts. Okay, let's quickly do a poll and see if you know what this one is. Go ahead. What is that? Good. Okay, very good. Almost all of you got it. It is a durovenous sinus where you drain excess CSF. Broca's area, we did that earlier. Okay, how about this one? Let's do this one as well. Okay, so this is the primary somatosensory cortex of the left brain. If you damage that, you will not be able to feel from the right side. Okay, that's the answer. Okay, stay the main problem associated with each of the following. Let's go through this one. Hydrocephalus, main problem. What's the main problem with hydrocephalus? CSF circulation is blocked. Okay, maybe cannot drain. Okay, so that's going to cause CSF to build up in the ventricles, resulting in high pressure in the brain. Not good. Meningitis. Meningitis is the inflammation of meninges. Okay, so you're going to have fever, you're going to have headache. It could be caused by bacteria or viral infection. Could even be caused by fungal infection in rare cases. Glaucoma. High eye pressure. Right, pressing on optic nerve. Okay, you might eventually go blind if the pressure is not being controlled. Astigmatism. Okay, the main problem is an uneven lens. Okay. Light scatters inside the eye. Okay. So the image is going to be blurry, especially at night when it's like raining. All the light appears to be fireworks. Okay. If you have astigmatism, you will know what I mean. If you don't and you find it hard to visualize, just Google astigmatism and then press Google image. It will show you what it looks like for those of us who have astigmatism. It's caused by an uneven lens. Presbyopia. Presbyopia is related to aging, and you lose visual accommodation. Okay, accommodation is when your lens is able to change the thickness accordingly as you shift the focus from far away to nearby. And what happens is those suspensory ligaments get a little bit, it's like an elastic band, right? You pull it many times, it loses the elasticity. So that's what happens when you get older and you cannot change the shape quick enough. So you get presbyopia. You cannot focus on what you're reading. Floaters, so you have debris, builds up in the aqueous humor. Cataracts is clouding of the lens, right? Because protein starts to crystallize. Myopia, eyeball is too long. Cannot see far. Hyperopia is opposite. Eyeball is too short. Cannot see near by. Okay. These are some of the things that we've done many times in class already, so you can go through it. Here's an extra diagram for you to practice. And you know, these are some additional multiple choice. I think you should be able to handle it yourself. Okay, if there is one that you're really not sure of, then you can send me an email. And then here is another diagram for you to label as well. Okay, so please take the time to finish the labeling. Okay, just before you go, let's just do another challenging question here for application. Okay, go ahead. Okay, anyone else? Okay, nobody got the answer, unfortunately. Okay, the answer is D. Okay, when you spin many, many times, remember those fluids in those tubes, once you stop, it will keep going a little bit, right? Because of inertia. And that is why it will affect the perception of your head position. Okay, because the fluid is still moving, even though you're not moving, the fluid will stimulate the vestibule and tells you that your head is still moving, and so you lost your sense of the perception. Okay, that's the answer. And here, one last one. We just talked about this a few minutes ago. Okay, I have one person who gave me an answer. Anyone else? Yeah, it's a very long class. I think a lot of you are running out of juice. The answer is reticular formation. Okay, that's the one that maintains consciousness, right? And so if you dampen that, you're going to be unconscious. Okay, that's it. A lot for you to do until our next class. As always, if you have questions, just send me an email and I will get back to you as quickly as I can. Okay, good luck studying and I'll see you all next week. Bye for now.